Hi guys, just bear with me a minute now. I get my class up. Sorry, just bear with me 1 second. Trying to share my lesson, which is not sharing. Okay, we're ready. Okay, so hello folks and welcome. So my name is Jennifer and I'm the biology teacher here at Grinds 360. So today we're going to do everything that's related to plant reproduction. And I've broken it up into different sections. So pollination, then I have male and female meat formation. And after each section, we're going to look at some past paper questions and answer those. So on our newsfeed, we should be able to download those past paper questions and work your way through this. So this is live, there is a chat. But will you be able to draw the diagrams for the meat formation? Yeah, so there is a chat here. There you can ask questions and I can answer them straight away. So we've already got a question, so we'll be annoyed. So you're not annoying me, Sarah, but thank you for your question. Are you able to draw a meat formation for questions? Yes, you can, and we're going to go through those. So thank you. Okay, so plant reproduction, you can see it comes up practically every single year. So last year there was the question in section B, an experiment question, and on section C. So there's a very high probability that will come up again this year in section C of your paper. So let's start off first. They might ask you to label the plant and functions of the plant. So your sepal, that we have our sepal down here. So the main function of your sepal is going to protect your flower and prevent it from drying out. Then you have your petals. The main thing with your petals, they're bright and they're colourful and they're there to attract the insects for pollination. And your reciprocal, so your reciprocal down here, so that's where your flowering plant arises. So there's two parts to your flowering plant. There's the female part and there's the male part. And they say with biology, if you're able to label a diagram, you're able to draw that diagram. So please be aware of that. We don't expect students to be artists. So how diagrams are corrected, we'll look at the diagram first. Does it look like what it's meant to be, even though you don't have to be an artist? And then we look at labels. So here we have the female part of the plant. The female part of the plant is called your carpel. Your carpel consists of your stigma at the top, your style all the way down here, and then our ovary. And then within your ovary, you have your ovules. So your stigma is where our pollen is going to land for pollination. Your style is where your pollen is going to travel down, and then your ovary contains the ovules where double fertilization will take place. And then we have our male part. So the male part is called the stamen. It consists of the anther, which produces the pollen, and then it has the filament down here, and your filament is going to support the anther. Now, the stages that we're going to look at today are pollination, gamete formation. I'll do, I think it's male first, and then we'll answer some questions on male and then female. Then we'll look at fertilization. We'll look at seed and fruit formation and dispersal and dormancy and germination. I'm not going to focus on the experiments within this chapter. Those who are on the platform know that I've done masterclasses on the experiments, so I won't be focusing time on them because I do have 2 hours to get this done. So let's look first at pollination. So you need to know the definition of pollination. So it's the transfer of pollen from the anther to the stigma of the flower of the same plant, if asked. Now, self-pollination involves the transfer of the pollen from the anther to the stigma of the same plant, and then cross-pollination involves the transfer of the pollen from the anther to the stigma of a different plant. So you need to show the direction of where that pollen is going, from where to where. Is it the same plant for self-pollination, or is it a different plant for cross-pollination? So something they may ask is advantages of pollination. So the main advantage of pollination is that it can prevent overbreeding. And it also, another nice advantage is it can bring about greater variation in species. Sorry, I have a question, my phone's down here. So is there notes to download? No, it's just no notes to download. The only notes to download page are the questions that I'm going to work my way through on this. And the questions are all past exam papers, and I'm going to show you how to get the perfect answer in those questions. So those questions are on our newsfeed. They should be there for you to download. So no notes to download for today. But if you're on the platform, those notes are there within the textbook that you're able to see. Now, there's different types of method of pollination that we need to know. So insect pollination is going to be with a flower, a nice colourful flower that has petals. So that can be insect pollination. You can have animal pollination, or we can have wind pollination. So for insect pollination, is just animal first. So with our flowers, they are brightly coloured petals, so that's going to attract the insects. The pollen is sticky, so that will stick on to the insects, and that stigma then is going to be inside the petals. Now, wind pollination. So wind pollination, it's usually like a grass, okay, or I have there oak. So with wind pollination, there is no nice brightly coloured petals there. With wind pollination, there usually is a very large stamen, and usually also the stigmas there also can be very large and feathery, and they're going to hang outside of the plant. The pollen though is very small and very light. So let's look at a question that was asked there in 2024. Sorry, let me just put my tissue over here. So in 2024, what is meant by the term? We're just asked for pollination. So the term they're asking for pollination, what we wanted to see from students was that it was the transfer of pollen from the anther. So I'm going to write this down as well alongside with you. So it's from the anther to the stigma. So what we wanted students had to have was from and to be awarded the marks there. So the transfer of pollen from the transfer from the anther to the stigma. The second question then students were asked were to distinguish between self-pollination, and distinguish between cross-pollination by writing a sentence about each. So you will come across the word distinguish, and if you're asked to distinguish something in the course of biology, you really are saying what is the complete difference between them both. So usually it will require a sentence for each, or maybe possibly a definition for each here. So if students are asked this, so I'm going to distinguish so the examiner knows, so I'm going to say self-pollination is pollination within the same plant, And what we were looking for here is we're looking for same plant to be there from students. And then with cross-pollination, so it's pollination between, sorry about my writing, but I'm just conscious of time, between different plants. And what we were looking for there was different. Sorry, once I've got another question to come through. Oh, yes, sorry. So Holly, yes. So Holly says, yes, there is a big typo in the book, in the textbook. There's a few typos in the textbook. So with going back, so the typo is up here. So within self-pollination, I have the word different. So fix your book, it's Holly, since you have the book there, and put in there same. Okay, thank you. Are these questions on exam papers? If so, what year? This question here was 2024. So that's, sorry, Killian's asking a question there. So there is the question. So I have all the questions that I'm going to cover today available for you to download. So I have stuck with past paper questions. Then we're asked part B, sorry, so that was a question from Killian. So then we're asked B part 1. And it's frozen. I'll fix that in a second. So I have to actually just charge my pen for a second. So I'll talk through this one. So with B part 1, we're asked to label the parts A, B, and C. And then we're asked to give one adaptation of this flower, visible or otherwise, that shows it is wind pollinated. So A is our anther, so there's 3 marks for that. B is our filament, and then C is going to be your stigma. That one caught a few students off. So then you're asked to give one adaptation of this flower, visible or not, that shows it's wind pollinated. So you could say that the pollen is very small. Another one is that we have a large stamen. Another one is that we have a feathery stigma. So let me just check. Pen. So I'm going to have to charge my pen there. Now, this one is from Deferred 2022. I wouldn't ignore the deferred papers from your studies. So there's 2 deferred papers that are there for students to look at. The Deferred 2022 and the marking scheme, and the Deferred 2023 and the marking scheme. So this was asked in the Deferred 2022 marking scheme. So draw and label the structure of the stamen of a flower. Indicate the letter P, that shows the pollen, the location where pollen is made. What is meant by pollination? So that was asked there in 2024. Distinguish between cross and self-pollination by writing a sentence on each. Asked just there in 2024. And name 2 ways in which cross-pollination can occur in flowering plants. Okay, my pen. I don't know. So let me I'm just charging this to see. So your diagram. Oh, there we go. I'm back up. So again, we don't expect students to be artists. So I am not the best drawer, but if I'm drawing this, I'm just going to make sure I clearly label. Another exam tip, guys, is a black pen. Everything you read in a textbook or a book is in black because it's easier on the eyes. Your papers are corrected online on a computer screen. Black is easier on the eyes for your examiners rather than blue. If I come across a few blue scripts, I actually had to take a step back, but it's much easier for the examiner to correct or to mark it with a black pen. So I'm just doing one line straight up, and from my line, I'm going to do that. Okay, so this is the stamen of the flower. So I'm going to label this. So I know it says draw and label, so it's saying draw and label the structure of the stamen of the flowering plant. So here I have my filament, and here I have my anther. So there are 2 main ones. Together, I'm going to let the examiner know, put this up here, that this is the stamen. So it asks, indicate with the letter P the location of where pollen is made. So we know pollen is made within the anther, so you can put P, an arrow coming out of P, anywhere there on the paper. Now, another tip with drawing diagrams, please don't do tiny little diagrams. If you want to do half a page of a diagram. I would also advise, I didn't really do it there, draw your diagrams in the middle. So that means if you are going to be labeling it, you can label from the left and you can label from the right. Another tip with labeling is, I'm saying here, this is the anther. Another tip, is, if I go back. I'm saying this is the anther here. If I do this one here as a label, it's not going anywhere. It's not pointing to anything. So make sure if you're drawing a line, it's going to directly what you are labeling there. Okay, so let me go back. Here is P. So then it's asking us what is meant by the term pollination. So that's the transfer of pollen from the anther to the stigma. Then we're asked distinguish between cross-pollination and self-pollination. So cross-pollination is pollination from pollination between the anther, between different plants, and then self-pollination is pollination between the same plant. So then we have named 2 ways in which cross-pollination can occur in flowering plants. So a few options here, which I'm going to put here so students can see. So cross-pollination can happen with animals, it can happen with insects, it can happen with wind, and it can happen with water. So there's 4 options there for you for that. So that is pollination. So we're now going to go on to male gamete formation first is what I'm going to look at. So with male gamete formation, so a gamete is haploid. You need to know the definition of haploid. Haploid is one set of chromosomes. Your gametes are sex cells. I'm just fixing this here. So we're going to look at our male gamete formation first. And Sarah did ask there at the beginning of the class, if asked this, could we draw a diagram to show this? And yes, you can. If you feel a diagram, a nice labeled, clearly labeled diagram will help you with this. So male gamete formation occurs within the anther. This is what we know. So this is going to be our pollen. So your anther consists of 4 chambers. Our sperm nuclei or our pollen grains. So they're going to be formed in each pollen sac. So when these pollen grains are fully developed, what will happen is the anther is going to shrivel, and if the anther is going to shrivel, then that's going to release the pollen. So within each pollen sac, we're going to look at how the male gamete occurs. So the male gamete consists of the microspore mother cell, which is diploid. So our microspore mother cell is going to divide by meiosis. So here on the side, I have a little diagram here explaining this as well. Now, when that divides by meiosis, we're going to form here 4 haploid cells. So you can say we can have 4 haploid cells are formed, or you can say a tetrad, which means the 4. This tetrad is going to split and it's going to form 4 young pollen grains. So that's what's happening here. So I'm just trying to make sure that this is all on. Sorry, just fixing this size. Okay. There we go. Now students can see. Okay. So our 4 pollen grains here. Now, so each one of these is going to be a potential male gamete. Now with this, our pollen grain development, what's going to happen is one of the nuclei here within this young immature pollen grain is going to divide by mitosis. So this haploid nucleus here is going to divide by mitosis. So we're going to end up with 2 nucleus. And one of those is called the generative nucleus and the other is called the tube nucleus. So to go back over, our microspore mother cell, which is diploid, is going to divide by meiosis to form a tetrad of pollen. So that means each one of these is haploid, if this is divided by meiosis. To remind you of diploid, 2 sets of chromosomes. So that's your link there back in with the genetics. So our tetrads, so our pollen, so these are joined together, and what will happen is they will separate. So each one of these is your young pollen grain. Each nucleus within the pollen grain here is going to divide by mitosis, and we're going to have 2 nuclei within that pollen grain. One of the nucleuses is called the generative nucleus and the other is called the tube nucleus. Now what we also need to know is we need to know, so I have here generative nucleus and tube nucleus. Now also on the outside, our outer layer, our X sign, this is our pattern for different specific flower species. To be honest, I've never seen them ask that. So when our pollen grain matures, like I mentioned before, the anther walls are going to dry up and split, and that is how our pollen grains are going to be exposed to help. Our pollen grains are going to help with pollination. So pollen grains are not the gametes, okay? It's the nuclei inside that's the gamete. So the gametes are going to be from the generative nucleus, which we're going to look at. So now let's look at a question on male gamete formation. So the pollen grains are produced in the anther. Outline the main events in the development of pollen grains within the anther. Usually with male gamete formation, it's usually 9 marks for a question of this. The magic number in biology is 3, so that's 3 3s. Sorry, I need a sip of water already. Another tip for answering questions in section C is you don't need to do like a full paragraph. You can do it in bullet points or a sentence on each line, because what the examiner is looking for, we're looking for some keywords along that, and we'll underline that. or put the tick beside that, and that's where the marks are awarded. It's easier reading down than going across, over, back, and that would be another tip I would tell students about. So how am I going to answer this? So I'm going to answer this in bullet points, and then I'm going to go through where the marks are going to be awarded for students. So our diploid, microspore, mother cell. I'm just going to do the division side here for time-wise, and this is going to divide by meiosis. That's my first point. My second point, I'm going to say when that divides by meiosis, it will produce 4 haploid nuclei. Or I'm going to put a slash here, say tetrad, because some people, you may have been told tetrad, both is correct. Okay, so the nucleus of each will divide by mitosis. 1 nuclei is the generative nucleus. The other nuclei is our tube nucleus. That's all we need to say if you're asked about the development of the male gamete formation. And if you're doing this and you're like, oh, forget which one is formed, so just remember here your events, in a sense it has here, outline the main events of the development of the pollen grains in the anther. So if you can remember that the anther is related to the male gamete formation. Another thing here, another word is outline. So outline usually is a minimum of 9 marks as well. So that means 3 points, 3 correct points. So while you just may know 3 of these, just learn 3 of these, I would advise learning all of it. So I'm going to highlight with a different color here of green of where the marks are going. What's on a scheme? What do we want to see students have? We want to see students say diploid. So there's 3 marks for diploid. There's 3 marks for saying microspore mother cell. There's 3 marks for saying divides by meiosis. There's going to be 3 marks for saying produces 4 haploid nuclei or tetrad. The nucleus of each will divide by mitosis. We'll have a generative nucleus and we'll have a tube nucleus. That's what we're looking for from our students. We are called candidates from the candidates. So if in an exam you had forgotten one of these and you had diploid, that was correct. You mightn't have said microspore mother cell, you might have said divides by mitosis. The examiner will still read through what you've put there to see can they give you the marks. We won't take away the marks, we're trying to give you the marks. So you could have stuff here in the middle that's wrong. That still doesn't matter, we have to read through all of what you have there to get the marks. So that was in 2022. Now if we look at 2020, again, which wasn't asked because of COVID, but they shared the paper with us and the marking scheme with us. Answer the following questions of sexual reproduction. Describe in detail. So we're seeing the word describe. That's a key word. So again, a minimum of 9 marks with the word describe. Describe in detail the development of the pollen grain from the microspore mother cell. So again, if you wanted, you could do a diagram with this because it does not say do not draw a diagram. Across biology, if you feel a diagram will help you with your answer, you're able to draw a diagram and making sure that it's clearly labeled. It'll help you with your answer. So the same thing, I'm going to do bullet points down again. So of pollen grain from a microspore mother cell. So microspore mother cell is already there. So we're saying here, the diploid, and we can say again, microspore mother cell. But there won't be marks for that because it's already implied in the question. The microspore mother cell divides by meiosis. We have here to produce 4 haploid nuclei. So each one of those are your pollen grains. So the nucleus of each will divide by mitosis, and it will produce our pollen grains. So it will produce pollen grain with 2 haploid nuclei. Actually, did I put that in there? Sorry, I made a mistake. So add, if you're writing this down, this sentence here, produce pollen grain with 2 haploid nuclei, and you forgot it up here by mitosis. So I'm going to write in here, produce pollen grain with 2 haploid nuclei. Actually, that should have gone there. Okay, so here's another example. I've added the sentence here, produce pollen grain with 2 haploid nuclei. If you read this through, it says diploid microspore mother cell divides by meiosis to produce 4 haploid nuclei. Then I have the nucleus of each will divide by mitosis, produce pollen grains with 2 haploid nuclei. That's not in the correct order, but you can still get marks for that if it's not in the correct order. So ideally, that should go there. So the better answer, in order, down here. So I'm going to say produce pollen grains with 2 haploid nuclei. 1 is going to be the genitive nucleus. Sorry, 1 nuclei. Is the genitive nucleus. The other, or you can say 1 nuclei, is the tube nucleus. Again, keywords, where are the marks going for? Diploid. Not microspore mother cell, because that's here, it's already implied in the question. But you can see in 2020 it wasn't there, so we can get the mark for it. Would I learn diploid microspore mother cell? Yes, I would. So diploid microspore mother cell divides by, microspore mother cell divides by meiosis. So 3 marks there. 3 points there, that's so far 9 marks. To produce 4 haploid nuclei, the nucleus of each will divide by mitosis, produce pollen grain now with 2 haploid nuclei. 1 will become the genitive nucleus, 1 will become the tube nucleus. So there's 1, 2, 3, 4, 5, 6, 7, 8. 8 points for a question that would be 9 marks. So we'll talk about the role of the genitive and the tube nucleus at fertilization, but at the moment we're just looking at the male gamete formation. So if they're asking about the development of the pollen grain, this would be your answer, the perfect answer for the development of the pollen grain of the male gamete. So now we're going to look at female gamete. So our female gamete formation. Just going to change my highlighter. So here we have our ovary. Okay, so here is your ovary. Within the ovary are the ovules. Okay, so here we have the ovules. So each ovule is composed of 2 outer walls, and these are your integuments. So at the end, we have a micropyle, so that's a small opening of the integuments, and that's where our male gametes are going to pass through. Your inner layer, each ovule has a nucellus, which we'll talk about in a moment, and that's where the developing embryo is going to get its nutrients from. So within each ovule are a number of diploid cells, one of which is going to become the megaspore mother cell. So that's what we're going to look at now. So your megaspore mother cell is going to divide by meiosis to produce 4 haploid cells. With the male gamete, it's the microspore, okay, that's going to mother cell. That also will divide by meiosis. Of this, so you could say where you have your 4 haploids, and that produces the 4 haploids, that's still the same as the tetrad. So you could say it produces the tetrad. That is still the same. So of these 3 haploids, 3 of the 4 haploids, 3 are going to die, okay, or degenerate. And 1, whatever the surviving one left over, that's going to become the embryo sac. So your haploid embryo sac, that is going to divide 3 times. Okay, so when it divides 3 times, we're going to produce 8 nuclei. 5 will die. We don't need to know about the 5 during leaving cert. We're just going to look at the remaining 3, 2 of which are going to be the polar nuclei and 1 is going to be the egg cell. So let's just look at this in a diagram. So this can also be asked in a diagram. I have a question. Have you ever been asked to draw the xylem tracheids? Yes, you have. They have been asked to draw the xylem tracheids. Actually, I'm covering that in tomorrow night's. If you are with the demand platform, I'm covering that tomorrow night's grind, plant structure and transport. But yeah, you have to be able to know to draw the diagram or the xylem tracheids and the vessels. Going a bit off topic. Here's your xylem tracheids. Showing your lignin as well. Okay, so back to your female gamete formation. So here we have our megaspore mother cell dividing by meiosis. And here we have our 4 haploid gametes. 3 of these will die. So you can say die or you can say degenerate. Our 1 embryo sac remaining is going to divide by mitosis 3 times. So that's important. So if it's, so that's going to divide by mitosis 3 times. So here we have then in your so we're going to have 8 haploid nuclei. We don't need to know about 5 of those. The 2 that we are concerned with is your, wrong color pen. The 2 polar nuclei in the center, which are all haploid, and our egg cell down here in the center, which is haploid. No problem. Did they ever ask you to show a female gamete formation? Yes, sir. So this is actually an example here that you can use this diagram here to answer you with the female gamete formation. Or you can write it out if you want. So another, I think I'll put a space here. So another way of drawing this is I have my microspore mother cell. So I'm just going to draw first, okay? Okay, so here is so that's my drawing first, and now I'm going to label. So here's my microspore mother cell, which is diploid, or megaspore, excuse me, megaspore mother cell. This is going to divide by meiosis. Here are my 4 haploid cells. I can say here, 3 die. So here I have my embryo. You can call this your immature embryo. This is going to divide by mitosis 3 times, by 3. Does that look like a 3? So the 2 polar nuclei that we're looking at, so there's 3 up here, 1, 2, pen's coming down, 1, 2, 3. There's 3 down here, 1, 2, 3, and there's 2 in the center. My 2 in the center are my polar nuclei, which are haploid, and the one down here I'm interested in is my egg, which is also, haploid. So that's your female gamete formation. So your megaspore mother cell is diploid. The megaspore mother cell will divide by meiosis to produce 4 haploid cells. 3 of these die, degenerate. Your embryo, well then the remaining embryo will divide by mitosis 3 times. In our mature, actually let me put this here, sorry. So this is important. I'm just going to call this, so this is your embryo sac. So here's our mature embryo sac. And within the mature embryo sac, we have 2 polar nuclei and our egg. So where are the marks going for this. What do we want? Our microspore mother cell, diploid, divides by meiosis. 4 haploid cells. 3 die, degenerate. Your embryo divides by mitosis 3 times. So you have to say the 3 here for this. And then within our embryo sac, we have our polar nuclei and we have our egg. So that is important. Now we're going to look at questions on female gamete formation. And I want to draw your attention that the female gamete formation has not been asked since 2019. Okay, so not been asked since 2019. It did appear on the 2023 deferred paper, but it was not used on the 2023 paper or on the 2024 paper. With female gamete formation, it's usually 12 marks for this. Okay, male gamete formation is usually 9 marks. For female gamete formation, it's usually awarded 12 marks. So then that's your 3/4. So this can be answered with the diagram. This can be answered with text. So I'm going to do both. I'm going to answer, so give the account of the development of the egg cell within structure Z, and you'll see what the other part of this question was as we go on this. So how are you doing with that? Okay. So I'm going to do this one in bullet points, and then in the next one, I'm going to do it as a diagram. It doesn't say here, do not draw a diagram. So I'm not going, so yeah, if you see do not draw a diagram to aid your answer, then don't draw a diagram. Or it might say a diagram may be drawn to aid your answer. So that means you can draw a diagram. You don't have to draw a diagram. So I'm going to do bullet points again. So I'm saying diploid, megaspore, mother cell. Divides by meiosis. And that will produce 4 haploid cells. 3, I'm going to say here 3 die, or you can say degenerate, whichever is easy, whichever suits you. Okay, so what we have left over, so our remaining embryo divides by mitosis 3 times. Our embryo sac contains 8 nuclei. 1 becomes the egg, and there's no harm letting you know that the egg is haploid. And then 2 become our polar nuclei, which is haploid. So where are the marks going for this? So I'm going to do that in green. So diploid, megaspore mother cell, divides by meiosis, produces 4 haploid cells, 3 die. Remaining embryo divides by mitosis 3 times. So you must have their divides by mitosis 3 times to get the mark there. Your embryo sac contains embryo sac contains 8 nuclei. 1 becomes the egg, which is diploid, and 2 become the polar nuclei. So if there's 12 marks being awarded for that, you have 1, 2, 3, 4, 5, 6, 7, 8. Again, 8 points for this. In 2019, when this was last asked, okay, 2019 was the last time female gamete formation was asked on the paper. Describe in detail the development of the embryo sac from a megaspore mother cell. So I could answer this here, or I can do a diagram, or you could do both. Some of you might prefer doing both. So if I do, but I do know that we will accept, actually when this was last asked, we accepted students do the diagram. So I'm going to start off with drawing my circles first. So 1, oh, my page is gone, give me 1 second. Here we go. So one circle here. I have 1, 2, 3, 4. Bracket these off. 1, 1. Line here, arrow there. Oh, doesn't want to do an arrow. Arrow here, arrow going across, making sure there's a nucleus in each one. Okay, so now I'm going to label this. So here I have my megaspore mother cell. Which is diploid, which is going to divide by meiosis. We have 4 haploid cells, 3 die. Okay, so our embryo sac divides by mitosis 3 times. I'm just going to draw this a little bit bigger. So then my mature embryo sac, we have 8 nuclei, 1, 2, 3, 4, 5, 6, 7, 8. So I'm going to call this our mature embryo sac. And the 2 nuclei I'm concerned about are here in the center, which is your polar nuclei, which are haploid, and then here, sorry, I'm running out of space. Let's say here's my egg, which is haploid. So where will you get the marks? Megaspore mother cell, or a diploid megaspore mother cell, divides by meiosis to produce 4 haploid cells. 3 die. Our embryo sac will divide by mitosis 3 times. And we're going to, in our mature embryo sac, we have 8 nuclei. So we can see here 1, 2, 3, 4, 5, 6, 7, 8, of which 2 will become our polar nuclei and 1 will become our egg. So that's all we need to say for female gamete formation. If asked, remind me the last time it was asked was 2019. So if you do your past paper questions, you'll see that this is part of a question of 15C. So it wasn't just there alone. 15C back in 2019 was 30 marks. So it's part of a 30-mark question. In 2023, it was 16A. 16A is now our 30-mark questions, your A, B, C, D. Okay, so it's part of a 30-mark question. So I don't know, I wouldn't ignore it. Claire, could you take me? Yes, Claire. So Claire's asked, could you technically get the marks before you even finish the diagram? Yes, so if 12 marks are going for this, and I'm correcting student paper, I would literally go, so if a student drew, so if a student wrote this, I'll go back to this one, so 12 marks for this. So I would underline, diploid, megaspore mother cell, divides by meiosis, produce 4 haploid cells, they've got the marks. I'll have a glance to see there, but I'm not going to spend time correcting that part here, this part here, if I've already awarded the student the full marks they can get for the diagram. Now, if the student makes a mistake here or something doesn't make sense, I will keep reading through, and your correctors have to read through there once and try and give you the full mark. If you were drawing the diagram, the same thing. I'm looking at the diagram and we underline, there's a red, like it's a red squiggly thing like that. So you will have an opportunity if you want to view your paper, and you should see a squiggly line like that, and that usually is where the student has gotten the marks. So you can see where you get the marks. So if I'm correcting this again in the summer and they have labelled megaspore mother cell, diploid, meiosis, 4 haploid cells, yes. And you can see from the diagram that they are correct. Maybe they might have labelled this and said, okay, so here for an example, if they don't have the word there, or they have haploid cells, we can clearly see underneath that there's 4 haploid cells in case a student made a mistake. If they didn't say died, that doesn't matter. I could still move on somewhere and see what's labelled. But it's very important with your polar nuclei that you are showing that there's 2 polar nuclei. Sorry, another question come through. Do you need to include? Yes, so Claire's asked that they're to prove to show that they're haploid. I would show that they're haploid, because usually a follow-on question from this can be about fertilization, and with fertilization, obviously we need to know that they're haploid. Usually the follow-on question with this is, where does fertilization take place, or explain the double fertilization. But I would put, Claire, I would put in the to show that they're haploid. So that's all they can ask us about female gamete formation. So the question in 2019 was phrased, describe in detail the development of the embryo sac, okay, from your megaspore mother cell. 2023 deferred paper, give an account of the development of the egg cell within structure Z. You'll see what structure Z is in a moment as we go through this. Okay. So hopefully that's a bit clearer now. You don't need reams and reams of sentences or paragraphs. Here I put them in concise, short bullet points for you. Which leads us on to fertilization. So if you're asked about male or female gamete formation, more than likely the question following that will be about fertilization, or where it occurs, or how it occurs, or what happens. So fertilization, what is it? So it's the fusion of the male and female gametes to form a diploid zygote. So here we have our female. So we have our stigma at the top where our pollen is going to land. We have the style where our pollen is going to travel down through, and your style will lead down to your ovary. Within your ovary is going to be your ovule. So your pollen grain is going to land on the stigma of the carpel, our female part. Our tube nucleus, so within that pollen grain, I'm going to take a sip. Our male gamete. So that pollen grain is our male gamete with 2 nucleuses. Okay. Our generative nucleus and our tube nucleus. Okay, so our generative nucleus and our tube nucleus, both which are haploid. Now, your tube nucleus is going to form a tube. That's what it's going to do. It's going to form a tube. So its tube nucleus causes growth of the pollen tube down the style. So the tube nucleus is going to go all the way down here, down to our micropilli. So the tube nucleus grows down towards the micropilli. Now, this is to do with chemiotropism. So we're linking in with plant tropism. So this tube nucleus is growing down a tube to chemiotropism. Chemicals are given off, so it knows which way to go. Once the tube nucleus gets towards the micropilli, it dies off, it disintegrates. The tube nucleus is there for the generative nucleus, because the generative nucleus, as it's moving down the style here, what will happen is this generative nucleus here will divide by mitosis. And when it's divided by mitosis, it's going to form the 2 male gametes. Okay? So your mature pollen grain is going to land on our stigma here. Here's our pollen. It's going to land on the stigma of the carpel. Within your tube nucleus, we have our generative nucleus within our mature pollen grain, we have our generative nucleus and our tube nucleus. Your tube nucleus is going to form a tube down the style towards the micropilli. What causes the tube nucleus to form that tube? Chemiotropism. So once the tube nucleus gets towards the micropilli, it's going to disintegrate. So it's basically growing down and making an entrance in here for our tube nucleus, for our 2 male gametes to enter. Our generative nucleus, as it's going down, so I'm going to rub out this here. As it's going down, this generative nucleus will divide by mitosis. So now in the red now, this is now my 2 male gametes. Now, I don't see them asking you to draw this, but maybe explain it. So these are dividing, this divides by mitosis. These are your male gametes. So if our tube nucleus, I'm going to do blue here, has formed this tube down here. Now my two male gametes can travel down here. So your tube nucleus, kind of like a wingman, it's bringing it, tip the nucleus, and it's going, okay, I'm gone now, see you. The rest is up to you. Right, so in come our two male gametes. Now, this is important. This is where double fertilization occurs. Double fertilization occurs within our embryo sac. What is double fertilization? So this is with our two gametes fertilize what's inside the ovule. One gamete will fuse with the egg to form our diploid zygote. That diploid zygote will eventually form the seed. The other male gamete will fuse with the two polar nuclei to become the triploid endosperm. So one of these male gametes will fuse here, if I zoom in, with our two polar nuclei. This will become your triploid endosperm. The other male gamete will fuse here with our egg. I'll do a different color. This will become your diploid zygote. So I'm saying 2N for showing diploid zygote. I'm writing here 3N. Where did the N's come from? One male gamete is N. We have two polar nuclei. I'm just going to write here polar, N, and add them all up with your triploid endosperm. Hi Jennifer, is the tube nucleus you drew in blue the only part of the tube nucleus that dies, or does it all die? That part will die. Eventually the others will, because, thanks, quite good question, Holly, because eventually what will happen is this will become the seed, will become the fruit, will become the new plant. So it'll all eventually die. Good question. I like that question. So our polar nuclei, our male gamete is haploid. Our two polar nuclei are haploids. That's your 3N. That gives rise to your triploid endosperm. And then our diploid zygote we have as our male, which is haploid, and our egg, which is haploid, becomes our diploid zygote. So what are they more likely to ask you about here? So they're more likely to ask you, it may ask you the role of the tube nucleus is forming a tube from the stigma down the side to the entrance of the ovule, to the micropilli. Now, even then, I don't think I've seen us want micropilli on a scheme. What is important is your generative nucleus divides by mitosis to produce your two male gametes. Then it will ask about double fertilization. One male gamete, I don't know which one male gamete will fertilize and join up with the diploid zygote to become the triploid endosperm. One male gamete will join up or fertilize the two polar nuclei to become the triploid endosperm. Did I read that right? I'm getting confused now. I'm going to say it again. One male gamete will join with the egg to become the diploid zygote. Yeah. One male gamete will join up with the two polar nuclei to become the triploid endosperm, which was asked last year. So we're going to look at the question that was asked about this last year, and I'm going to show you the marks there. So two fertilizations occur during sexual reproduction in flowering plants. Describe what happens during both of these fertilizations. So we're going to say here, I'm just going to change my color pen because I want to stick with black. Okay. Oh, I have to charge my pen again. So, sorry, bear with me. Right, so I'm going to go back to this sentence here. So of 2024, it asked, describe what happens during both fertilizations. So we wanted to see one male nucleus joins up. Now, did they accept sperm? They did, but there was a big debate about it's not really sperm. Okay, so one male nucleus or one male gamete joins up with the egg to become a diploid zygote. Now I'm going to be, what we wanted at first was we wanted a male gamete and the egg for 3 marks to become a diploid zygote for the second 3 marks. But they weren't getting that, so then they accepted male gamete, 3 marks, egg, 3 marks. If they didn't mention that, but they said diploid zygote, 3 marks. So they increased the chance for the student to get the mark. Which I would strongly advise, if it asked this in 2025, male gamete and the egg for your 3 marks forms your diploid zygote, your second 3 marks. So that's 6 marks. Then your other 6 marks, one male gamete and 2 polar nuclei. And it's very important that you mention that it's 2 polar nuclei. For your 3 marks, first 3 marks, and the second 3 marks forms your triploid endosperm. So I'm going to have to wait till my pen get back. So that was last year. Now the question before this was, I believe, male gamete formation. They wanted to know how the male gamete formation was. But they haven't asked female gamete formation since 2019, was on the 2023 deferred. So leading on to this was describe the formation, or describe the main events that occurred in male gamete formation or pollen formation. So I think that, well, yeah, that was 9 marks, and then this followed on here for 12 marks. 3 marks for one male gamete, one egg, double for diploid zygote. 6 marks, 6 marks then for male gamete, 2 polar nuclei, and forming a triploid endosperm, 6 marks. Now, I want you to bring your attention to 2023 deferred paper. So we've answered the female gamete formation part here, but let's see this question as a whole, okay? Sorry, a little bit of nose. And I'm going to have a sip of water. Check the time, it's 3 o'clock, great. So the diagram shows the carpel of a flower. A pollen grain is trapped on the stigma, so we can see here, and it's showing us the carpel. Name the parts labelled X, Y, and Z. So I'm going to read through the question first. So it's 16A, it's in 16. I know every question in 16 is going to be worth 30 marks. So here's your choice of A, B, C, and D. Another thing, a tip regarding when you look at your script and when you look at your mock, go and have a look what's in 16 and 17. There might be some really nice questions there in 16 and 17 for you to answer. In question 16 and 17, you're required to answer 2 of A, B, C, or D. Now, if you answered A, B, and C, your best 2 are picked. But if you answer A, B, C, and D, your best 2 are picked also. My other tip regarding this question, that's a massive pet peeve of mine, is for each question, do a brand new page. Okay, so I would say that to my students: new question equals new page. There's nothing where I find it's, as an examiner, I'm doing it from my selfish reasons. As an examiner, it's so much easier to correct one question on one page instead of then a student having another question directly under squashed in. The booklet is so huge, your answer booklet is so big, there's so many pages there. So it's much easier to correct it that way. Can you leave out human reproduction? You could, if you wanted to. It was all about the male last year. I suspect it'll be about the female this year, but you could leave reproduction out. Last year reproduction fell in question 16 or 17, I can't remember, I think it was 16. So it was a 30-mark question. Prior, see that, it can be a 60-mark question. I think one year they mixed human reproduction in with plant reproduction. Just be aware of that. That too, but it's up to you. Is that Quiva? I think it is. It's up to you, Quiva. Now this says give one other way in which pollen can be carried to the stigma of a flower. Copy this diagram into your plants. Copy this diagram into your answer book. and draw a pollen tube to show the pathway taken by a male gamete. The pollen grain has a tube nucleus. Describe how this nucleus gives rise to the two male gametes. Give an account of the development of the egg cell within structure Z. So that's the question we answered previous. So here's the rest. And then I did say before, usually they ask after male gamete formation or female gamete formation, they ask about the double fertilization. And this leads us on to two fertilizations occur during sexual reproduction of flowering plant. Describe what happens at each fertilization. The question, is plant guaranteed? Long question. Plants will be there. Yeah. Or they could put all the plants together as a 60-mark question. They did last year, and it was a lovely question. Actually, students did really, really well on it as a 60-mark question, and I had an awful lot of candidates get full marks in that plant question. Don't be scared of plants. Students can be rewarded for, not many students answer plants. So students can be rewarded for answering plants. What do I mean by that? If they have to front load, so instead of being 3, 3, 3, 3, 3, 3, 3, if they have to front load because we're if they have to front load, they may, and they have in the past, gone to plants and front loaded in plants. So you can be at an advantage of answering plants. I like teaching plants though, but I know not everybody likes plants. But another thing, just keep looking over the past paper questions, and hopefully that will help. Let me see my pen ready. Yeah. Okay, so let's look at this deferred question. And what was asked here that wasn't asked in 2024 or hasn't been asked in a while. So it starts off with name the parts labelled X, Y, and Z. So you are going to put in your answer book part 1 X, Y, Z. So X is your style. Yeah. My brain's gone. X is your style, Y is your ovary, and Z is the ovule. Another thing, guys, if they're saying to you label X, Y, and Z, make sure you clearly show what X is, not Y is. Don't leave it up to me to figure out what your X, Y, and Z is and just write stigma, style, ovary, whatever order. It's clearly label. So it's asking you to give one other way in which pollen can be carried to the stigma. Sorry, the pollen grains can be carried to the stigma by animals. What's a other way? So your other way can be, so for the sake so you can see the question, could be wind, or I'm going to put here as an option, or water. Now you're asked to copy this diagram into your answer book. Again, reminding you, you don't have to be an artist really good at drawing. I told you, and I'm going to say again, I'm not good at drawing. So if I'm drawing this again here, okay, so this is part 3. Okay, there's my diagram. Okay, make sure I have my ovule in the center. So that's my diagram. It says copy the diagram into your answer book and draw a pollen tube to show the pathway taken by the male gametes. So here is my pollen. So draw a diagram. Here's my pollen tube. So I'll just get a different color pen for this. So I'm just going to draw literally a line, come down, go around. Oh wait, I don't want to go all the way down there. I'm going to draw and I'm going to show them that this is my pollen tube. And I'm going to label in here my scent, or just draw on here my egg cell. Next question, sorry, I'm going to have to go back up. Give an account of the development of the egg cell by the female. So we've gone through that. Then we're asked two fertilizations occur. Oh no, I skipped a sentence, sorry. Which was not asked last year or a couple of years. The pollen grain has a generative nucleus. Describe how this generative nucleus gives rise to the male gametes. So what does the generative nucleus do? That as it's going down, so our generative nucleus is going to divide by mitosis. So for you here, I'm just going to write this. Okay, so our generative nucleus Actually, you don't even need to say genital nucleus because it's already implied there in the question. Genital nucleus will divide by mitosis. Now, could you just leave mitosis on its own for that? Yes, because that's what we're looking for there is mitosis. Then we have given account of the development of the egg cell within structure Z. Now, do we need to draw a diagram? Yes, you could to draw a diagram for that. So just to recap back over this, we have is our given account, we have our diploid megaspore mother cell divided by meiosis, produces 4 haploid cells, 3 die off. Okay, we have our immature, our remaining embryo divides by mitosis 3 times. Our embryo sac contains 8 nuclei. One is the egg cell, which is haploid, and we can just put N behind that. The other is our 2 polar nuclei. Our 2 polar nuclei, haploid. So that was that question there we answered. Now if I go back to the last part, 2 fertilizations occur during sexual reproduction in flowering plants. Describe what happens during each fertilization. So that's where my pen went in the other one. So let's look at this. So we can say one male gamete fuses with the egg cell. I can say here it's haploid, and this will become our diploid zygote. Now you could also say, literally say 2N diploid zygote. So the marks for that will be a male gamete, oh, wrong colour pen, sorry. A male gamete fusing with the egg to become our diploid zygote. So the marks for that should be 2 3s. Okay, 3 marks for your first male gamete in the egg, 3 marks for forming your diploid zygote. The second part is we have one male gamete fuses with the 2 polar nuclei, and that becomes our, when there's space on the slide there, our triploid endosperm. Or you could say 3N endosperm. Where are my marks going for? Male gamete fuses the 2 polar nuclei, 3 marks, to become your triploid endosperm, 3 marks. Now that's the main chunk of our fertilization. So we've looked at male gamete formation and answered questions. Excuse me, sorry. We looked at female gamete formation. We've looked at fertilization. What can they ask us about fertilization? Which we've answered here, and then they can ask explain double fertilization. So hopefully you're prepared for that part if they ask about that part. So that can be worth, as we've looked at with the male or the female. The male's gamete formation, 9 marks, but that was asked last year. Female gamete formation can be worth up to 12 marks. It usually is 12 marks. Then if it asks you about explaining the double fertilization, that can also be 12 marks. There's 24 marks for you there. I think that's 8% there for you there. Okay, now we also need to know what, I think I have another question, do I? Yeah, sorry, I'll do one. So the other question here, 2 fertilizations occur during reproduction in flowering plants. Oh, sorry, I did that. That was the one I didn't. Yeah, sorry. Apologies. Okay. So we now need to know fertilization has occurred. So now we need to know what can occur after fertilization. So we're going to look at seed and fruit formation and sorry, just checking with the other what's the dispersal. Okay, so that's what we're going to look at now. So you do need to know what happens to each part of the plant after fertilization has occurred. So your zygote becomes your embryonic plant. Okay, so that was our diploid zygote. That's going to become the plant, your embryonic plant. Your endospore sperm, our triploid endosperm. That's going to supply food to the developing embryo. Your ovule will become the seed. The ovary becomes the fruit. The ovary wall becomes the wall of the fruit. The integuments become the testa. So you do need to know what becomes what after fertilization has occurred. So what is common? So you should know that your zygote will become the plant, the future root and the shoot. That's your developing embryo. Where does that get food from? It gets it from the triploid endosperm. So the triploid endosperm will become, after fertilization, will become the food supply. The ovule will become the seed. What's the seed? Think of the seed like a seed on strawberries. Your ovary will become the fruit, strawberry, they're red. And then your ovary wall become the wall of the fruit, and then your integuments will become the testa. So after fertilization, we're going to look at that now. So after fertilization, our diploid zygote grows repeatedly by mitosis, and it'll form the embryo. Okay, so it's going to divide by mitosis. So your embryo consists of your plumule. Your plumule is your future shoot, your radicle also, which is your future root. The cotyledons as well. So the cotyledons, so here's my embryo here in the purple. Your cotyledons are your food stores for germination. Your triploid endosperm will repeatedly divide to form the endospermic seed. So this is going to act as the food store for the developing seed. This brings us on to two types of seeds. So this is linking back to where within plant structure and tissues, you'd have to learn the difference between monocots and dicots. Okay, monocot, one seed leaf, dicot, two seed leaves. Your monocotyledons and your dicotyledons. Then you know the difference between monocot and vascular tissues, structures in stems and in stems in roots. So that's their TS of a dicot stem. Why do we use dicot? Because the vascular bundles are neatly arranged around the side. In a monocot, our vascular bundles are scattered. You would have looked at leaf venation, and another maybe example is number of petals on a flower. So now we're going to look at the difference of seed type within a monocot and a dicot. So monocot means one seed leaf if asked. Our monocots are going to store the food in the endosperm, and they're classified as endospermic seeds. What does that mean? So that means they contain some of the endosperm when they're fully formed. So in my green here, I'm saying that this green is my endosperm. This purple is my embryo. So if I go back up here to show you here my green, here is my endosperm. What's your endosperm? Is your food store for the developing embryo. In the purple, that's my developing embryo. Your developing embryo consists of your plumule, your radicle, and your cotyledons. So your monocot, one seed leaf, and they store food in their endosperm. They're considered endospermic seeds because they contain some of the endosperm when fully formed. Your example, grass would be an example, of an endospermic seed. Dicot means two seed leaves. Okay, so here, there's a difference if we look. So here, if I go back to this diagram here before I scroll ahead. In my monocot endospermic seed, I can see here I still have some endosperm, and here is my embryo. In a dicot, you can see here it's all purple. Okay, so that means that endosperm is all gone. So most dicots, they use up their endosperms before the seeds mature. So they're known as non-endospermic seeds. Okay, so here is all of this really here, guys, is your embryo. Now I'm going to scroll on to the next page. So a reminder that the ovary becomes the fruit. The ovary develops into our fruit. Your seed will form our ovule. So the ovule is what can release growth regulators. So that's linking back in with plant responses. Now, advantages or functions of the fruit they will have are to protect the seed And when the fruit, another thing is for the fruit to attract the animals. We want the animals to eat them, and they'll disperse of these and pass them away. And what it's doing is passing the seed away from the parent plant. So it's spreading whatever that plant may be. So we're going to try and colonize new areas. Now, seedless fruits, these are naturally occurring seedless fruits, which means they are genetic varieties, so they do not produce seeds. So for some examples, bananas, some grapefruits, some oranges. Artificially induced seedless fruits can be formed by spraying plants with growth regulators like auxine for your seedless grapes. Ethane gas can also be used as well to ripen bananas, tomatoes, degreen lemons, and oranges. So that's your seed and fruit formation, which leads us on to seed and fruit dispersal. So dispersal, what does it mean? It's the transfer of the seed or the fruit away from the parent. Know that definition, please. Okay, so you can say here option, you can say seed or fruit away from the parent. So that's what we want to see. That is the transfer away from the parent plant. So why is dispersal necessary in plants? Okay. So what it can do, apologies, it can minimize competition, it can avoid overcrowding, colonize new areas, and increase chances of survival. So I would know at least 2 reasons there why we need dispersal. What is dispersal? What's an advantage of dispersal? What's a benefit of dispersal? So different methods of dispersal and adaptations. So we need to know a method for wind, animal, self, and water. So there are your 4 different types of dispersal it could ask you about. So if I go through here for wind, I have our little sycamore seed, helicopters, you might have played them when they were younger. Another example would be dandelion as well. So they are winged and they're going to be taken off by the wind. They're going to spread that seed. What are they doing? They're minimizing competition, they're avoiding overcrowding, they're colonizing new area, and it's increasing their chance of survival. Animals can be carried, so fruit with hooks can cling to animal fur and get carried away. So example there are burdock, nature's velcro, which is the long sticky plant you might stick on someone's back when you were younger. Sticky fruits. Animals is another example. So animals, what they can do is eat the food, digest it, and passes through them. What's the point of dispersal? Minimize competition, avoids overcrowding, colonizes a new area, increases chance of survival. An example of self-pollination or self-dispersal can be beans or pods, or peas, excuse me. So what will happen is the pods will dry out, and when it dries out, it splits and it will spread the seed, the food. What's the purpose of dispersal? Well, what is dispersal? The transfer of the seed or the fruit away from the parent plant. Why is it necessary? What's the advantage of it? Minimizes competition, avoids overcrowding, colonizes new areas, and increases chance of survival. And then your last example here is water. So with water, your example would be water lily, and they have air-filled fruits that float on the water, and that disperses them down the stream. So now let's look at a question that could be asked with seed and fruit formation and dispersal. So again, where's my question? Okay, I'll go back to dormancy in a moment. I'll just put this in the wrong one here for the moment. Okay, so bringing you back again and showing you deferred paper 2023. So this is also question 16. So in question 16, we looked at question 16A about the female gamete formation and fertilization. Now we have, that was 30 marks. Now here's another example of a 30-mark question that could be asked, that possibly could be asked. So in the diagram shows 2 seed types, A and B. So I'm going to read through the question, then we'll answer the questions. So is the seed in diagram A classified as a monocot or a dicot? Give a reason. Copy either seed to your answer book and label each of the following parts: the tester, the cotyledon, or the embryo. Name one example of a monocot seed and one example of a dicot seed. Which part of the embryo is the germinate in a germinating seed gives rise to each of the following: the root and the shoot. Answer the following questions in relation to seed dispersal. What is meant by dispersal? Name two methods. State one advantage. Okay, so again, nothing like this has been asked for a while. So let's look and answer this question. So which type of seed in the diagram? In the seed diagram, is the seed in diagram A classified as a monocot or as a dicot? So here's the diagram here. I scroll back just a couple of pages where I had this diagram in a picture, and we can see here that a monocot has more endosperm, but the dicot has no endosperm. So monocot or endospermic, dicots are non-endospermic. They've used it all up. So we go back here. So bear with me now a second. Here we go. So we can say A is a monocot. Okay. Then it's asking me, so that's still not enough for my answer. It's asking me to give a reason for this. So we can say that there is endosperm. We can see the endosperm. We can see the endosperm is not all used up. And just sorry, not, and you can see some of this. It's not all used up. So we can see some endosperm. So we've qualified our answer. You could also actually say, because technically down here, this is the embryo part, you could also say it's a one seed leaf, because that's what a monocot is. But I'll go up there with your endosperm, not all used up, it's visible. So copy either seed into your answer book and label the following parts: the testa, the cotyledon, or the embryo. Okay, so I'm just going to copy here A, number 2. And again, I'm going to put a line through here. Okay, it's telling us to label the testa, the cotyledon, and the embryo. So my testa here, I'll try and do both. Your testa is here for your monocot. Your embryo, all this, like you can even just, let me just, for us, what I'll do is I'll just color this in. So all of my green part, this is my embryo. Now also within here would be, remember we said with your embryo is made of your plumule, your radicle, and your cotyledon. So anywhere where there is green, we can say that this is our cotyledon. Have I spelled that correct? Okay, conscious of time. So then you're asked part 3. I'm going to put part 3 here. Obviously, you will not do this in exam, but for the sake that we can see part of the question and the answer. Name one example of a monocot seed and one example of a dicot seed. So I'm going to say monocot seed, for example, could be grass. Dicot seed, I can't choose, my mind's gone blank here. What did I say as an example? They're broadbean, yes, sure. P. There's some question there. P, on the P. So then your answer, I'm going to go underneath here again so we can see. So let me scroll up so you see part of the question on the answer. Of the embryo in a germinating, which part of the embryo in a germinating seed gives rise to each of the following parts? So number 1, the root. The root will become the radicle, to think about that. And the shoot becomes the plumule. If you forget about that, or root radical, okay, your plumule is what's going to go and grow above overground. And then we're asked, let's put here, dispersal. Dispersal not been asked for a while. So What is meant by the term dispersal? So we learned back up here, dispersal is the spreading of the, or the transfer of the seed or fruit away from the parent plant. Sorry. When it's space, I'm going to say away from the parent plant. Name 2 methods. So, sorry, that's the part 1. Oh, sorry, can you? Part 2. Name 2. Sorry, let me go back up. Name 2 methods used by plants to disperse seeds. So we know of 4. So we could say wind or water or animal or cell. That's all it's asking is to name 2. Say 1 advantage of dispersal to the plant. So it can be less, number 3 I'm going to put here. So colonization avoids competition. So I'm going to say avoids competition. Colonize new areas. Increase chance of survival. Okay. So dispersal hasn't been asked for a while. So that's your seed and fruit dispersal. Now, if we go over, and what we'll do is, and we're going to finish on this, how we're doing time-wise, checking, half an hour, great. So what we're going to do now is we're going to finish on, no, I have to go back. We're not finishing yet. We're going to do dormancy, and then we're going to look at germination. So we're going to answer dormancy and germination, and then do questions on that. So you know, that's a good one. So following dispersal, what can happen? The seed may go through a period of dormancy. What is dormancy? Dormancy is a period low metabolic metabolism when hard and tongue-tied. Okay. Dormancy is a period low metabolic is a period of low metabolic rate and cell activity. Okay, so that's what it is. So when the active growth is suspended, that's what it is. It's a period of low metabolic period of low metabolic rate and cell activity. That's what it is. So a seed will fail to germinate when conditions are suitable. What can cause this? So growth regulators can cause dormancy. So the presence of inhibitors prevents the emergence of the embryo, or we have a lack of promoters prevents growth, or there may be like an impermeable testa. Advantages of dormancy. Okay, so I have 4. I'm going to say no 2. Your advantage is it's allowing time for the embryo to mature and ripen. That's 1. We can increase the chances of dispersal. We can survive survive unsuitable conditions, and we can survive. Seeds will germinate when conditions are suitable. So no, so that it may be phrased as what's the advantage of dormancy? What's the benefit of dormancy? So that's why they wrote there. So special conditions. So how do we break dormancy? Special conditions can be needed to break dormancy in agriculture or in horticulture. So what could be doing for that? So the seed may be soaked in water. Now, when it's soaked in water, what that does is that softens the testa. So that's linking back with the experiments. By soaking it in water, it softens the testa. So if it softens the testa, what it does is it activates the enzymes. So it's going to activate the enzyme lipase, amylase, and protease. They're the enzymes. So soaking it in water, the benefit of that, linking back with the experiment, is it activates the enzymes. We can expose it to light or dark or cold temperatures for periods so that it will germinate, depending upon what the seed is. Now, there may be reasons for dormancy. So the testa could be too hard to allow water in. The testa could be too hard to allow the embryo to emerge. So our embryo needs time to emerge, and growth inhibitors can delay it. So this is linking back to the triploid endosperm and the diploid zygote. The triploid endosperm is your food store. So and then our diploid zygote is our developing embryo. Your embryo is your radicle, your root, and the cotyledon, your future shoot, your future root. The cotyledon, whether it's going to be a dicot or a monocot. Your triploid endosperm, that's your food store. So if we soften the testa, so the testa is hard, the embryo cannot get the food from the endosperm. To develop the food from the endosperm, for it to develop and it to grow. For it to grow, we want the shoot to grow up above the ground. Okay, once it has leaves, it's then going to provide its own food. If we soften the testa, we allow water in, it activates the enzymes. So therefore, the developing embryo can break down and use those use the nutrients that are there. So reasons: testa too hard to allow water in, testa too hard to allow the embryo to emerge, to develop. So the embryo needs time to emerge, and core growth inhibitors could be there for that. Now, the importance or knowledge of dormancy. So it allows, and this, again, the phrasing of this can be linked in horticulturists, farmers, agriculture. What's the knowledge of dormancy here? So the knowledge allows for optimum storage, allows for optimum ploughing or sowing, sow the seeds when it's the right time, and it allows seed treatment. So that's sacrificing. So that could be breaking the seed. And what you're doing here, you can see the line, you're actually softening the testa. Why do I want to soften the testa? I want to activate the enzymes. And by activating the enzymes, it's going to help the developing embryo to grow. So I've known these two reasons for that. Now, this brings us on to then germination. So germination is the regrowth of the seed into a new plant. And this will happen after a period of dormancy when conditions are suitable. So what are the conditions needed for germination? We need oxygen, a suitable temperature, and water. But why do we need oxygen, a suitable temperature, and water? We need oxygen, which allows for cellular respiration. We need a suitable temperature, so it's the right temperature for the enzyme activity. Okay, so that's the enzymes within our triploid endosperm or endosperm. And for water, we need water to activate the enzymes. Water will soften the testa, activate the enzymes. So water is going to be absorbed and provides a medium for chemical reactions. Now, you think back already to your knowledge of enzymes and respiration, those two chapters. So if I have the seed just there on its own, it's not going to respire, it's not going to grow. So the water is of utmost importance to soften the testa, to activate the enzymes. We know that enzymes are involved in respiration. Okay, we know what can affect the active site of an enzyme, an unsuitable temperature. If the temperature is unsuitable, the substrate will not join up with the active site of the enzyme. We're not going to form a product. So hopefully you see the link there. So we have water first, activate the enzymes by softening the testa. Okay, so we've activated the enzymes and now we have a nice suitable temperature. So the suitable temperature will cause chemical reactions to occur. One example of the chemical reactions is cellular activity. So this is where we have glucose being made in the presence of oxygen, will help to form CO2, carbon dioxide, and water and energy. So there's a link there. So this is really important. Now, events in germination in a seed, which is just sort of what I talked about there, is the seeds absorb the water and the oxygen. So your enzymes digest the food stored, okay, within the cotyledons or the endosperm. The nutrients are going to be transferred to the growing embryo. So what are the nutrients? So the nutrients could be glucose, amino acids, fats, and glycerol. So these are the products. What was the substrates? Glucose from starch, amino acids from proteins, fatty acids and glycerol from lipids. What's the enzyme that's involved within this? So glucose, it's an amylase. With amino acids, it would be protease, lipase, with fatty acids and glycerol. So I'm linking that back in with our knowledge of enzymes and our knowledge, as we could say, human nutrition, or our knowledge linking back in with food and digestion. So your nutrients are transferred to the growing embryo. New cells are used using amino acids. Okay, remember new amino acids as well. It's also involved in DNA formation. And our glucose can turn into ATP, adenosine triphosphate, and that can also for an importance with the cell division. So nutrients are used in respiration to make energy and in the formation of new seeds. Our radicle will become our root, which will stay underneath. Our plumule will become the shoot. And then what will happen is the shoot will eventually here grow up, okay, which leads on to the next one. And then what will happen is photosynthesis can now occur. The plant can now photosynthesize. So now there's no more involvement now. So the plant is autotroph. It's going to make its own food through the process of photosynthesis. So now linking in another chapter in with this. So digestion contributes to successful germination as it makes the nutrients soluble. So cotyledons, what happens with cotyledons is they can remain below the soil. Your example is a broad beam, so that's your dicot. Cotyledons can move about the soil, example sunflower seeds, so that's your monocot. And again, I'm showing it there with your leaf venation. Now I've added this in here, and I'm going to have a question. I'll do a question on this, just checking my time. So changes in dry weight of a germinating seed. So if you have the questions, I have this blank and I just typed it in there. So here we have dry weight, dry mass of a seed. We can see here on our y-axis, on our x-axis, we have our time, so going in 12 days. And we can see we start off with our seed here. You can see here we have part of our embryo. Okay, so from your embryo now we can see our plumule, our shoot. We can see our radicle, our root. After a number of days, we can actually then, our plumule, our future shoot, our radicle, our future root. After a number of days, we can see our roots, and we can see here we have got a shoot, okay, and the shoot has got leaves. So looking at this, days 0 to days 9, the mass decreases. So here we have our seed. How am I going to activate the seed? I'm going to soak it in water. What will that do? It'll activate the enzymes. So now it can help to start to germinate. So now the seed can absorb the oxygen, it can respire. Okay, so it can it can respire. Glucose, amino acids, fatty acids, and lipids, they're now made. So now our seed can develop. So as our nutrients are being consumed by the embryos from days 1 to 9. So that's why there's a decrease in the mass of the seeds. What also is taking place is respiration is taking place. So there's now also going to be a loss of CO2. And the water content can now vary between the seeds as it's developing. So I'm going to leave that there for you to write in. So that's important. So what's happening then from days 9 to past to day 12? So our mass is increasing. So your mass of the seed is increasing because you can see here, and still keeping that up there, there's now leaves there. So what is the plant doing? How is it making its own food? By photosynthesis. So I'm going to put this up and just let you take that down and fill that into the box or screen grab shot and take a picture. So what you're doing there is you're explaining the mass of the seed over a period of time. So really qualify your answer there. And over time, that mass of the seed will not be the same because the plant is now making its own food through the process of photosynthesis. And the last part, hopefully I have it down, we're going to look at and compare the embryo and the cotyledon mass as well. So, changes in dry weight of the cotyledon or the embryo of one seed. So the dry mass again on our y-axis, time here on our x-axis. So we can see from day 0 to day 4, 6, 8 that the dry mass of your seed here is increasing, and of my cotyledon it's high on day 1 and it's decreasing. So what is happening here? Your embryo is now digesting the food stored within the cotyledon. It's using the food stored within the cotyledon. So the mass of now your cotyledon, that's why the mass is decreasing as its food is digested. So what we're explaining here is this part here and this part here, this crossover. Sorry, excuse me. We can still see that our embryo mass is increasing as it's using up all those nutrients. And cell respiration. Respiration is taking place. We're making lots of energy. And if we're now making energy, what we're able to now to form is new seeds. So that developing embryo will form the plumule, or future shoot or radicle. And that's going to use up all that cotyledon, and eventually, my voice is going. Eventually, it's going to form your shoot, and the roots will stay down. And now, once the shoot is broken through the surface, it's going to be in contact with light for it to photosynthesize that way. So let's look at a question on this. And the example that I'm going to use is 2015, that's here. And it's more explaining, it's this, what I'm, the mass that I'm looking at that I want to answer. I'm getting tongue-tied now. So, where is my 2015? I don't have it. Sugar. Well, okay. So if you have the booklet, do I have a blank page? Right, I have a blank page. So if you have the booklet, I have my question here. So I'm going to talk through the question. You have the question in front of you in the booklet, and I'm going to talk through it. So part one is, if I'm just going to read here, sorry, that's why I'm looking over here, I have the past bit of question. So seeds can be classified as either endospermic or non-endospermic, based on whether food is stored in the endosperm or the cotyledon tissue. Seeds may also be either monocot or monocotyledons or dicotyledons. Following fertilization, from which part of the flower is the seed formed? And the seed is formed from the ovule. So I'm just going to write here ovule. Number 2 says, what is meant by the term monocotyledonous? What is meant by the term monocot? Monocot is one seed leaf. The third question. Name 2 biomolecules stored in the endosperm or the cotyledon tissue. So the word biomolecules there, we're linking back to food and biomolecules that you would have done at the beginning of 5th year. So instead of naming any 2, so I'm just going to put down our 3. We have proteins. We have our carbohydrates. And we have our lipids. Number 4 says, if one way knowledge of seed dormancy has been useful to humans. So it could be how we store and sow our seeds. That's what I'm going to write down. How we store and sow seeds, or when sowed seeds. Now, this is where I'm raging. So we have the graph. So the graph below shows the changes in dry mass of both endosperm and embryo tissue of a germinating endospermic seed over a 3-period week after sowing. Dry mass is the mass of a tissue with its water content removed. So that diagram is kind of like this diagram here, okay? So if the graph is a bit like that, so if I just draw it, actually that's better. I just draw in the graph. So just to show you here. So time, days. So 0 to 21, mass, dry mass in grams. Okay, so my embryo, I'm going to do my embryo in blue. It goes like this, and then it goes up, sorry. And then I'm just going to go red with my endosperm. Or this could be your cotyledon. Okay, so we're asked to describe this. So it has here, suggest why the measurement of dry mass is performed in these investigations. So your main thing here is that the variable here is water, okay? And the content of that water can vary the mass. And where did I get that information from? From, in a bracket there, dry mass of a tissue with this water content removed. So I'm going to say here, the variable water, or the water content, let's cancel that. Could vary between the seed. And that we know that that's getting used up. And also if it's respiring or developing embryo, it's going to be producing water. So that's how it can vary. Number 2 says, explain why the dry mass of the endosperm tissue decreases over the 3-week process. So explain why the dry mass of the endosperm decreases. So why is this red line going down? So what's happening? Our food is being consumed. That's the main thing. The food, the nutrients, is being consumed, used up by the embryo. Now the question there says explain why. Another thing that's happening here is, I'll scroll this up a bit, respiration has taken place. So we have a loss of CO2. Your third question states, what process begins at A? So what's happening here? Sorry, A was the embryo. What's happening here? So this is now shooting up. So now our embryo is now making its own food. So the process that's happening here is photosynthesis. And question 4 says, would you expect the total dry mass of the seed, our embryo, plus our endosperm to remain the same up to A? So would this stay the same up to A? And the answer to this is, sorry, I was writing A. The answer to this is no, as there is loss of mass due to respiration and a loss of CO2. So as loss of mass due to respiration and CO2. Okay, folks, so that's a lot of plants done with 10 minutes to remain and 9% on my iPad. So look, any question, please ask. Hopefully I've made things a lot clearer for you and you have or grasped hopefully a better understanding about plant reproduction, gamete formation, fertilization. We've gone through key questions. I've given you key points of how to answer these questions. I've shown you where things have not been asked for a number of years that may be asked, maybe in your mock or the actual leaving cert itself. So if you any questions, please ask. I'm going to stay here for another couple of minutes. And if not, hopefully you have a nice afternoon. I do hope it was useful for you. So some of you that have joined today that may have joined for free, hopefully you found this interesting. I do live grinds with my 6th years every Monday where it's interactive like this as well, where students are able to type in questions and I answer them back with the answer, just like today. And anyone that does join in the future, they can catch up and look at all the live grinds that have taken place. And then we have short on-demand videos as well. A new feature that I've added for 6th years that is only on the on-demand is that I have flashcards of keywords and definitions. Just exam keywords and definitions. I've added quizzes there as well. And then there's another thing I've added, I've forgotten. But there's a lot more content there for 6th year students. So hopefully that helped. I'll wait around for another minute or two. If anyone has any questions, I'm just going to have a sip of water. So I don't think any are going to come in. So I'm going to say, have a nice evening. If anyone does come in after I turn this off, I'll still be able to see. But have a good evening. Thank you, folks. And some of you that are on the platform, I'll see you again soon. Some of you that have just joined for today, maybe I'll see you again in the future. So thank you. Bye-bye.